Okay, hello everyone. Can you all hear me okay? All right. Well, for those of you who know me, I'm Anjali Goswami. I'm the president of the Linnean Society, and it is my great pleasure to welcome all of you here tonight for our talk by our speaker, Professor Uma Ramakrishnan. So for those of you who don't, are you all, is anybody here not a member of the Linnean Society? Okay, all right. Well, for those of you who are newcomers to the Linnean Society, this is a society that is obviously has been around for 236 years and is really dedicated to bringing together people who are interested in the natural world from its understanding and valuing to its protection. We are a very broad tent. We have scientists like myself, we have artists, historians, naturalists, amateurs, really anybody who's interested in nature in general. Is welcome here. And so if you'd like to learn more about the society, please do reach out to myself or any of the staff who are around or to the members who are here tonight. We'll be happy to tell you more about it in hopes that you will join with us in working for nature. So tonight, we are very lucky to have Professor Uma Ramakrishnan joining us. Uma is a professor at the National Center for Biological Sciences in Bangalore, India, where she has been since 2005. She got her bachelor's degree in physics, chemistry, and mathematics, and then a master's in biotechnology before conducting her PhD work at the University of California in San Diego and a postdoc at Stanford University. Now, Uma's research is incredibly broad in terms of the systems that she works on, but it's fundamentally looking at the processes that drive genetic variation, looking at things like rodents and birds, but in particular, tigers. Her work has been very important both from the pure scientific perspective, but also on the applied level, where her work has contributed to things like decisions about highway planning and highway building in India, so as to not disrupt the movement of wildlife. Now, her work is incredibly important and world-leading. She is certainly one of the world's leading conservation geneticists, and this is reflected in an incredibly long list of awards that she has won, and I'm just going to list a small amount of these here. She's been a Wired Innovation Fellow, a Kavli Frontiers of Science Fellow from the U.S. National Academy of Sciences, a Bass Fellow at the Field Museum in Chicago, Parker Gentry, she's won the Parker Gentry Conservation Award, a Fulbright Fellowship, a Wellcome Trust India Alliance Senior Investigator Award, the Beacon Award by the Society of Conservation Biology, and obviously, with all of these awards, it's no surprise that she was also elected to the Fellowship of the Indian National Science Academy in 2019. So I hope all of you will join me in welcoming Uma for her amazing talk.
So hi everyone, I'm Uma, and it's really nice to be here, a real honor to be in this space, which we all know is a very important one for the study of nature and evolution. I think, it's really important for us to acknowledge that, biodiversity matters. We all love biodiversity, we all love nature, and it's not just because it's beautiful, but also because the air we breathe, the fruits we eat, the water we drink is not possible without biodiversity. And today, this biodiversity is under threat. We keep reading about this, we lament about it, but there's various drivers, like pollution, habitat loss, all kinds of things which are driving, the loss of biodiversity, increasing extinction risks. And if you look at this even in terms of popular science, what you read in the newspaper, 1 in 4 species of, say, mammals is threatened with extinction. So this all seems very glum and very depressing. But as scientists, the first question you ask is, how can knowledge, how can understanding of processes, of what's happening around us impact what we do in the future? So then, as a biologist, then, what is it that drives extinction? So things like pollution, global change, these are, not, they are things happening to populations, But intrinsically, populations go extinct when they are very small. So small populations have high chances of extinction, and you can see that here in this figure. Basically, this is data from butterfly populations, and when they are small, we have high probabilities of extinction. On the other hand, when populations are in small patches, again they have high probabilities of extinction. So on the right, you can see these simulations which suggest that populations which are small and isolated tend to go extinct much faster. A third component which might be important in extinction, now the jury is not out on this, there's still a lot of active research going on in this area, is genetics. And this is actually a very recent paper on many butterfly metapopulations in Finland, where they were actually able to show from these data that populations that had higher genetic variation, and you can see that the differences in genetic variation are not a lot, but populations with higher genetic variation had lower probabilities of extinction, whereas those with low genetic variation, despite having more populations, still had high probabilities of extinction. Now, that's against a correlate. So, why do populations which have low genetic variation go extinct? Well, they tend to be small, and when they are small, stochastic processes or chance events result in fluctuations in what genetic variation is there. This is a process called genetic drift, and this can cause not such adaptive or not very good mutations or variations to increase in frequency, and that results in a loss of fitness and results in extinction. Similarly, more obviously, populations which are small are usually made up of relatives. And so when you have mating between relatives, you can have inbreeding and the associated inbreeding depression. We all carry in our genomes, in our DNA, bad mutations. And if we are related, we carry similar bad mutations. So overall, genetics can also impact extinction. So I'm from India, and let's see how India has been changing over the last 300 or so years. So basically, this is India in 1700 and India in 2000, and you can see that basically forest cover has gone from 60% to 9% of land area, and agriculture and villages have increased. So human footprint on the land has increased from 20% to almost 80%. And in fact, only 5% of India is protected, for wildlife, for nature. And this is much below the globally recommended, level, which is about 15%. So, of course, as a biologist from India, the first question I want to ask is, it's a space with high human population density, and some species have already gone extinct, famously like the cheetah, which is now extinct in India, the Indian cheetah. But there still are a lot of species around, there's still a lot of biodiversity. So, which species might go extinct and where might they go extinct? So, of course, a good system to look at this is tigers. And this is a really nice picture of a female. She's in a park in central India, and you kind of wonder what's going to happen to tigers in India. So tigers are a good system to study extinction. This is the global range of tigers. In light red is the historical range. So this is not a species which is very specialized, lives only in old growth redwoods or anything like that. They live all the way from the Russian Far East and tundra to tropical rainforests, right? So very generalist, large distribution, lived in over 14 countries across the world. But today, the distribution is about 5%, arguably maybe 8% in terms of land area of what it was in the past. And you can see in dark red are kind of the patches where tigers still exist. So tigers themselves have gone extinct in South China, in the Caspian area, the Caspian tiger, and of course also in island Southeast Asia. So we've already seen extinction of tigers, and so it's obviously very interesting to ask, will tigers go extinct in India? Why, where, and can we do anything about it? So basically, what I'm going to do today is to try to split the talk into these two. What are we going to do about it is kind of conservation, and why, how. These are kind of scientific questions. So we start with science, understanding what's happening with tigers, populations, and whether they're connected or isolated, because connection and isolation are currencies in which extinction operates, right? And then basically ask, how can we think about sustaining connectivity? And that's conservation, that's kind of more action. And I hope I'll convince you that basically future survival then depends on both these legs of inquiry, science and conservation. So you might wonder how we actually study tigers. They're really big and quite rare. So luckily for us, tigers are territorial. So they, in our forests, they walk on these, forest routes, and basically they mark the territory. So they defecate and they spray urine to mark, and so that other tigers can smell and say, oh, this is not my territory, this is someone else's territory. And we basically follow their routes. We collect fecal samples, and because feces passes through the intestine before it is deposited, it has tiger cells in it. We can get DNA from those cells. We also use other types of non-invasive sources like shed hair and saliva, and I'll tell you a little bit more about that as I go along. But basically, we go to the field, get samples, bring them back to the lab, extract DNA, sequence bits of information from it, and try and understand these processes as they're playing out. During the time I have been a scientist, there have been some revolutionary changes in technology. So, of course, genetics has a long history, and we're here today. But Mendel and Darwin thought about genetics. They thought about evolution, but they didn't know what it was that allowed evolution and genetics to play out. And it was only with the understanding of the structure of DNA, that's Watson and Crick and Rosalind Franklin, that we were able to think of how to read DNA. And it was in 1977, not so long ago, that Sanger spent a lot of time figuring out, once he knew the structure of DNA, what chemistry would allow us to actually read it. And it was only later that we could actually work with small amounts of DNA. A technique called PCR, or the polymerase chain reaction, allowed us to convert a droplet of DNA into a swimming pool of DNA. And then it was even easier to read it. But it was still something we had to read word by word by word. And it was only in 2005 when there was a massive technological change, and we had a new technology called next generation sequencing, where we could actually read many, many, many words at the same time. So it was parallel processing. And this basically changed the cost of sequencing. We all lived through COVID, and you saw how we were sequencing COVID genomes nonstop to understand how the virus was evolving. So basically, while we think of these technologies as really good in medicine and so on, it also is really good for us who work with small amounts of fragmented DNA, torn up bits of DNA, which we tend to get from feces or hair and so on. So basically, this change in technology also allowed us to study tigers and read their DNA better. So the first question we ask then is, what do tigers look like across their range? Tigers still have quite a large range, and are the tigers in the Russian Far East different from those in India? And what these are basically samples of tigers from across their range. We read their entire genome, which is 2,400 million letters long, and we compared these. A genome is like a book. We compared these books across tigers, and what we found was, of course, that Indian tigers, called Bengal tigers, are more similar to each other than they are to tigers in the Russian Far East, and so on. What's more interesting for us from the perspective of this talk is when we actually looked at genetic variation stored in these subspecies or populations of tigers. Indian tigers have way more genetic variation. So you can see the pink line, just even when you add more and more samples, it keeps going up. So that's the private or the unique variation present in Indian tigers. So that's really interesting that, despite having a lot more, say, for example, compared to Russian Far East, you keep having more and more variation. So that tells us that as a whole, tigers in India have the most amount of genetic variation. But the graph on the right shows us individual tigers and how, and the higher the bar, the more homozygous or, identical sort of an individual tiger is. So while together all Indian tigers have high variation, many Indian tigers are also inbred. So this kind of gives us a hint of the fact that, that figure I showed you earlier, how there's been so much of fragmentation and change in habitats in India. While there were lots of tigers in India in the past, now they're kind of sequestered, and this might have led to this, signatures of inbreeding that we see. If you look at tigers within India, there seem to be large areas, for example, the orange, right? The orange, or whatever, it's not circle like thing, where many populations of tigers seem to be genetically cohesive. Whereas other places, for example, the purple, the one where it's a single population, but everyone is pretty much very, very similar to each other. So, one, which is actually Rajasthan, Ranthambore, is already an isolated population compared to two, which seems to still be connected. Despite the fact there are millions of people there, there still seems to somehow be connectivity or movement of tigers in that area. So how does that happen? How is it that, in two, which is a large area, we still have movement of tigers? So we tried to study this. We basically collected samples from across this area, and this is my student Prachi, PhD student, exceptional student, all of the people whom I've worked with. I'm lucky to work with some amazing people. Walked thousands of kilometers, collected these tiger scats, brought them back to the lab, and basically looked at how much of genetic variation was shared across these landscapes, and then asked, okay, if there's a lot of sharing between two populations, what is between them? Is it forest? Is it agriculture? Is it roads? Is it cities? What is between those two areas? So this is called landscape genetics, where we can infer whether specific landscapes facilitate movement or are providing resistance to movement. And what you can see on the right is what's called a resistance map. So that's basically how tigers see that same landscape. The darker the color, the harder it is for them to move. The lighter the color, the easier it is for them to move. And this is not a map of the roads, but what you can see pops out immediately are the roads. Those are national highways, and those blobs are cities. So basically, despite the fact that there's not a lot of forest or natural habitat in this large area, the main things which obstruct tiger movement are high traffic roads and high density human settlements. So tigers are okay. They're not unable to move if there's no forest, but they are unable to move when there are high density, like human impacts on the landscape. What about extinction? I kind of baited you all by talking about extinction in the beginning. Well, what we see in this landscape is that extinction is a function of population size and connectedness. So on the y-axis, we have isolation. The higher the isolation, the less connected the population. So small isolated populations on the top left-hand corner go extinct more often. That's why they're in red. Whereas large and connected populations don't go extinct as often. And in the middle over here are populations which are kind of small, not so small, kind of connected, not so connected. And their fate kind of remains uncertain. What happens to them will depend on whether they grow larger or whether they lose connectivity in the future. So this all sounds very nice, but it's kind of obvious. We just, if I did not put in, if Prachi and I didn't put in so much time and do all of this work, you might have guessed this. I mean, so what? So what is so interesting about it? But what we try to do next is to predict the future. So we asked, suppose this landscape changes in the future, what happens to these tiger populations? And here's one example of a future, let me see if I can, scenario. And this is when, suppose the parks are all fenced. Okay, so we don't allow any movement, that is, connectivity is gone to zero. And you can see that by 2100 many of these populations are now red. That means they have a high chance of going extinct. So what that tells us also is that what we see today as a large connected population or area is only because tigers are able to move. Once their ability to move is stopped, there will be extinction. So, so the good thing was, like I said, and Anjali mentioned, at the time we were doing this work, there was a big highway which was being constructed, and you can see these two parks here. On the right-hand side is what's called Kanha Tiger Reserve. On the left-hand side is Painch, where Rudyard Kipling wrote Jungle Book. Okay, and this highway, which was a 2-lane highway, was being expanded to 6 lanes. And we did the same simulations, the same approach to show, or we predicted what will happen if this highway now becomes a barrier. Because our data, our scientific information suggests that it would become a barrier if it's a wide highway with a lot of traffic. And what we found was that extinction probabilities increased by 50% in that landscape. So this was used in court. So basically, I mean, we didn't go to court. A citizen filed a public interest litigation. It's a legal tool in India. And they presented this paper and some other evidence, and they said, by building this highway, you're going to disrupt a natural process or movement of tigers from one place to another. And the court ordered the National Highway Authority of India to build this overpass for the traffic. So that's pretty cool that actually it went into conservation action. It was scientific data. It was a paper, a research paper. Most people don't read research papers, but it actually went and was deposited in court as evidence. And the really cool thing is that today, this mitigation structure works. So these are camera trap pictures from under. I mean, the underpass looks really ugly. I wish they would vegetate it a bit. But anyway, like trash and stuff lying around. But still, the animals, look at them. The mother and cubs, 2 males, these spotted deer, the gaur, the Indian bison, and dholes, Indian wild dogs, all moving from Kanha to Painch and back just because of this underpass. And had that highway come up without it, the connectivity would have been disrupted just like that, increasing potentially extinction probabilities. So I'm kind of saying that I want tigers to move. We want them to move through this landscape. So let's see what happens actually when tigers do move through these landscapes. This is again a nice montage of camera trap images of this female. She has 3 cubs. You can see they're very tiny, maybe a month or two months maybe. This is 9:15 p.m., 9:31 p.m., 7:40 a.m., and the same spot, 12:45 p.m. So basically, we're saying tigers should move, there should be connectivity, but then we're pushing them into places where there are people, right? And basically, like, I don't remember the number, but India has tons of cattle, right? So basically what's going to happen, these tigers are going to start livestock depredation, and that's what we see. There's livestock depredation, there's also human mortalities, and basically once there's this buildup of negative emotion against this carnivore, there may be retaliatory killings, poisoning, and so on. So there's a lot of tolerance in India, I have to say that. And this is becoming an issue because now 40% of India's tigers live outside the protected areas, the space which is allocated for them. So you must be thinking, okay, that's nice, but she said she works on genetics, so what is she going to do about this? So it turns out, hopefully you'll watch this video and see, this is a lovely camera trap video of 2 cubs. So they're like, these are actually cubs, they're like about a year old. But see what they're doing, they're eating this cattle. And they're chewing on it. This guy, he's not even bothered about the camera. Just looking straight into it. So tigers are just like blown up cats, actually. They're behaviorally very similar. But what you see is this cat is really handling the prey. And when it's handling the prey, what is it leaving on the prey? It's DNA, right? So that's what we did. We basically treated each of these kill sites as sites for forensic investigation to understand, about this negative human-tiger interactions. So basically, that's a jugular bite mark. Tigers kill at the jugular; they bite at the jugular. But you can see there's saliva there, there's shed hair, all kinds of stuff. So any kill site has lick marks. Cats always clean their prey. And then there's bite marks, and then there's shed hair, and sometimes there's fecal samples. And what we did is, it sounds crazy, but we went to many of these kill sites. We took like a swab. We all were familiar with swabs during COVID, and we just kind of pushed it into those bite marks and hoped for the best. Brought it back to the lab and said, well, let's see what we get. This is again work by my student Himanshu. Crazy field work. You're crouching over a carcass of a cow with maggots, and the tiger is probably somewhere around because you're at its food, right? So anyway, so we basically worked in two parks. That's Bandhavgarh and this is Kanha. They're both in central India, and we sampled something close to 145, 142 cattle kills in Bandhavgarh. Kanha was kind of more of a pilot. But first of all, the question is, does this work? Can we get DNA from these swabs or these lick marks, just like rubbing something on a carcass? And can we actually identify species and identify the individual, right? So basically, this is just some models, but basically they show that at least at 86% of the kill sites, we were able to identify at least one individual. And basically, of course, the number of samples we needed varied by the season. The monsoon was really bad. Most of the DNA was probably getting washed off, but it was the best in winter and a little bit worse in summer. And actually, shed hair worked pretty well. So the hair from the individual who was eating seemed to work well. Saliva sometimes was variable, basically the lick marks and so on. So now the question is, what did the results show? We were able to identify individuals. Were there problem individuals? Was there one tiger running all over the place killing cattle, and just taking that individual out would solve the problem of livestock conflict? Unfortunately, like most things in nature, it's not that simple. So this figure shows the number of individuals found at one kill site, two kill site, three kill sites, etc. So if there was a problem individual, one tiger running all over the place, we should have seen one individual at 8 kill sites, 10 kill sites, 15 kill sites. That's not what we see. We see almost 40% of the tigers of Bandhavgarh are indulging in cattle kill. So that means it's an ecological response. There's plenty of cattle around, and they're easy to kill in some sense compared to ambushing something like a deer, right? The cattle just sit down when the tigers chase them. So it seems very much like an ecological response. So how are we going to solve this problem? There are also some spatial pinch points, so areas where there's more cattle kills. Now that may be something one can address, maybe something to do with there being more cattle there, something like that. So there's a very famous case, in fact, of a tigress who actually killed people. And finally, so India, you're not allowed to kill anything in India. No, you can't hunt anything by law. But finally, the government issued a shoot order for that tigress. And you can see here, the tigers are such a big part of public imagination. There was a Bollywood movie based on that tigress. Her name is Agni. And there are all these people, young people protesting that in the cities saying she should not be killed. So clearly this is not just something which scientists are mulling about, but there's a lot of public interest in this. It's become something really, really important. How do we deal with this conflict? Not just livestock depredation, but people being attacked. So also I should say that, I kind of boasted that, we were able to identify a tiger at 86% of the sites, but basically we were only able to identify its DNA. I don't know physically who that tiger is, right? So management of the situation actually require me to tie that genotype or the genetic ID of that tiger to a live individual. And this is something actually that people are doing in New Zealand. These birds are called kakapow, and they basically made a database of the genetic signature of all the kakapow. And they basically, can actually identify, just from a DNA sample where individuals are moving, what they're doing, and so on. Now, this may not be feasible for all tigers. There's many more tigers than kakapow, but maybe it's something we can think about in actually taking that science on the ground into management. And there's also now, for example, people in San Diego Zoo who have developed in-field methods. So basically, in the field, you can actually identify individuals within a few hours. Like we had, those rapid COVID tests, for example, similar kinds of technologies. So there's still a long way to go, but I think there is maybe hope for some progress here. Okay, I'll tell you quickly a story of an isolated population. I told you that these populations are connected, but this is a story of an isolated population, and it's a population called Similipal, where for a long time the local tribals who lived there spoke of a black tiger, who looked like a ghost. They said. Now the everyone else, the forest department, the police, etc., just dismissed them, saying, oh, they're just making it up. Until in 1993, a youth actually, with a bow and arrow, shot in self-defense a tiger, which, I don't know, looks kind of black, right? It looks black to me. And this was the first proof that this very different phenotype, like you must have seen white tigers maybe in zoos, but this is a black tiger actually known only from this population. Once camera traps became more popular, these guys started showing up more and more, and these are how they look. Really, really beautiful. And it seems like it's a very rare phenotype found commonly in one location, and that immediately suggests that it's an isolated population. So, of course, the first question was, oh, why does this tiger look this way? Is it just something to do with its food, or is it actually some genetic variant, right? So, to answer that question, luckily for us, that's a very tough question to answer in a wild animal, which you barely see. But luckily for us, in a zoo, there was the birth of this litter, which included some black tigers, white but black tigers. So, this is a white tiger, but it has the same kind of pattern as a black tiger. And we were able to put together a pedigree where we again sequenced the whole genome and we looked at a specific gene and we found a single spelling mistake of just one mutation which was present only in the black individuals. And we did some analysis which suggested this is definitely the reason why it looks this way. We went back to the field, we collected samples, we brought them to the lab, we identified tigers, we identified individuals, we looked for the presence of this mutation, and we found that the frequency of this mutation was 60% in this one population, which is weird, right? So maybe we missed it elsewhere. So we looked again outside and we said, okay, maybe we didn't look for this mutation. Maybe it's there, but since it's a recessive trait, you have to have 2 copies of the mutation to look black. Maybe we missed it. We looked at 400 individuals, but the mutation was nowhere else. So then we went about asking, okay, is this a small and isolated population? So the answer was yes. Genetically, Similipal, this population we're talking about, seems isolated from the others which are close by to it. Also, individuals here are related almost on the order of siblings, suggesting again inbreeding, which would again lead to an increase in the frequency of this mutation. So basically, there were only 12 individuals we identified from all our samples. So Similipal does indeed seem to be isolated and small. So then we asked, well, we looked earlier at connectivity, right? There's our old populations, Kanha, Pinch, where the highways, etc. What if we modeled connectivity? Oh, sorry, I pulled this off. Does it seem like, there could be connectivity to Similipal? So it turns out that only if there is very, very high levels of dispersal on the order of 1,000 kilometers, that on average, tigers are moving 1,000 kilometers, would they actually get to Similipal? So it is simply too far away. And that's what happens, right? We're talking about this process of isolation. So this population already is too far away and it's already isolated. So we did some models to ask, okay, how could it be that this frequency is so high here? And those models suggest that unless the only way it could be so is if you had a very strong bottleneck where only 2 or 3 individuals survived and the population then recovered. The story continues. So we went, this was the 60% data was from 2018. We went back one tiger generation later in 2023, and already the frequency has gone from 60% to 75%. And should this population be left this way, it will definitely fix and everyone will be black. Now, that in itself may not be bad, but the fact is that obviously there's been a bottleneck, individuals are inbred, so this population is susceptible to inbreeding depression and future genetic effects. So, what do you do when there's something like this? I mean, you can't magically create connectivity, right? You can't magically create populations close by where tigers will disperse. Well, there's a concept called genetic rescue where maybe you can bring in individuals and allow them to breed in this population, increasing genetic variation, and hopefully this ticking time bomb may not go off. So we suggested this actually to the managers, and they took us seriously. And for the first time in India, a translocation of tigers was approved for genetic replenishment. And here she is, this one of the tigresses who was translocated to Similipal from very far away, and she's actually settled down. She and another female, and hopefully they will breed in the next few years, and there will be kind of new genetic variation introduced into this population. But the question is that isolation seems to have been building up over time. So how has this happened? How did Similipal become isolated in the first place? The only way to know that is actually to go back in time. And well, yeah. So there's been a lot of hunting of tigers for a long time. Trophy hunting was, oh, sorry, this got cut off a bit. But this is a picture, this is an Indian painting from a long time ago, and it basically shows the Mughals hunting tigers. Look at that tiger; it's so out of proportion big. But there's a huge hunting party here. There's even a bear over there, and also trophy hunting, where not just trophy hunting, but bounties were given out by during the British times for tigers that were hunted because they were treated as beasts and dangerous animals. So these are just some other pictures, but basically, there's many famous pictures and paintings of famous people who have hunted tigers. And this, for example, is a female hunted in that isolated population, Ranthambore in Rajasthan, by the Queen herself in 1963. So it turns out, though, that, oops, again, threw it off, that in spite of being so old, DNA actually survives. I'm sure you've seen in the newspaper about the sequencing of the mammoth genome, and Neanderthals are some of our archaic humans. So why not try? We thought, why not try to get DNA from these old and long dead tigers and see whether they can answer questions about how this isolation built up, what happened in the last 200 years, because it's all happened in the last 150, 200 years, not that long ago, not as old as woolly mammoths or anything like that, right? Very recent. So that's me actually sampling from a site in India. It's an old palace which actually burnt down. But can you imagine? I don't know whether you can see, but that trophy looks terrible, in terrible condition. But we are actually able to get DNA from it. And so here's another picture of a trophy room. So you can see there's about 8 tigers in this room. This is a small town in India where, one of our collaborators who's shared this material with us, his great grandfather actually hunted these tigers and all of these other animals. Wolves, hyenas, leopards. It's quite amazing, the collections that there are. So basically, we'd like to understand more about these tigers who are no longer with us. And so far, we have very simple questions. What was variation like in the past? What was connectivity like? Can we detect signatures of what's called purging, which is, initially when you have changes in population size, all of the bad mutations or the bad genetic variation gets cleared out. And one of the suggestions is that once that happens, populations should be fine. They should be able to survive. And can we actually use this to suggest strategies for conservation? So we've already collected about 175 tiger trophies. You can see from a spread across India, and about 70% of them are actually giving us DNA, which we can actually use to sequence the tiger genome. One of the reasons I'm here, this is actually in the UK. I'm hoping to collaborate with Katrina Guszczanski from the University of Edinburgh. She works on ancient DNA. She's done a lot of very amazing work. And I'm hoping that we can sample both trophies and museum collections from the UK. So in fact, what's really interesting is they might be a little bit older. A lot of the tiger trophies we've sampled are post independent India, post 1947. And until 1972, when the Wildlife Protection Act formally protected tigers and it was no longer legal to hunt them. So this is just a plug. If you know or if you own a trophy, it would be really wonderful to hear from you, because museums do have material, but this material is just sitting in people's houses, and if we can actually use it to understand something about the past, that's pretty phenomenal. So basically, the larger questions still exist. Uh, we started out by talking about extinction and population size and gene flow. These two things, gene flow is, connectivity. And somehow for tigers to survive in India, I think this gene flow has to be sustained. And this will unfortunately require managing development because, roads are needed. We can't just sit in Bangalore or Mumbai and say, oh, well, there's no need for roads in other parts of India, right? That's not okay. At the same time, there is also conflict. There is livestock depredation, people are being attacked, and all of those conservation problems will have to be managed. In some cases, intervention through genetic rescue may be necessary. But in the long term, I'm hoping we can move to having kind of stepping stone models where we have populations connected by little small populations where tigers can move, maybe breed, and then move on. Because otherwise, if there are these massive barriers like cities, there'll be death traps and they'll keep getting trapped and there won't be effective movement. So, in summary, that's a cartoon someone drew of me with a tiger some years ago. But, clearly the Earth is severely impacted by humans and we are witnessing species extinctions. I hope I've convinced you that genetic studies can help us understand species better and are important for conservation. But I think we're really just scratching the surface. We really don't understand why animals make the decisions they do. Why do they move? Why do they not move? Why do certain individuals look a certain way? What happens with inbreeding? I think we're just really scratching the surface of our understanding. But I think I don't want to sound very like depressing. I think there's a lot of hope. Like if there's a possibility for wildlife to survive in India with more than 1.4 billion people, we have the highest tiger population in the world. 75% of the world's tigers live in India, and they've actually recovered in the last 15 years. The population has almost doubled. So there's hope. I think in general, animals are very resilient. They survive. I think that we just need to kind of give them a chance and help them out a little bit by understanding a bit more about them. And I think that it's really important to understand that, especially in this space, that science and evidence is critically important, to facilitate recovery. Of course, by protecting habitat, we would be fine. But I think it's still important to understand how to facilitate recovery and to continue to monitor populations going forward. So, like I said, I'm really lucky to work with an amazing set of people. This is, my lab recently, and their friends and relatives. I'm very lucky to have many funding sources because this is expensive work. And I leave you with this painting from Odisha. It's called Patachitra. It's a folk art, but commissioned, which I commissioned, because the artist has put in camera traps and these black tigers and so on. I don't really noticed, but this is DNA along the sides. So thank you so much for listening, and I hope I finished on time. Yeah, and.